Vitamin C works through a variety of other mechanisms, such as blocking glucose uptake, which is really important in terms of cancer, generating hydrogen peroxide, and inhibiting viral replication. Most cancers rely primarily on glucose to generate energy through the process of glycolysis. Consequently, many types of cancers have increased glucose uptake. As I mentioned earlier, vitamin C is transported across cellular membranes by sodium-dependent vitamin C transporters and glucose transporters. The glucose transporters primarily import dehydroascorbic acid, the oxidized form of vitamin C, into cells. An in vitro study suggested that the import of dehydroascorbic acid into cancer cells can cause oxidative stress and decrease cancer cell viability. Some of the glucose transporters have a greater propensity to transport dehydroascorbic acid into cells over glucose. As a result, high concentrations of dehydroascorbic acid derived from vitamin C might inhibit glucose uptake into cancer cells and therefore hinder cancer cell fuel utilization needed for survival and growth. While vitamin C acts primarily as an antioxidant at physiological concentrations of approximately 50 micromoles per liter, pharmacologic doses of intravenous vitamin C greater than 1 gram generates hydrogen peroxide, a type of reactive oxygen species that can damage DNA, RNA, and proteins, leading to tissue damage. Multiple studies suggest that high-dose intravenous vitamin C can assist in cancer cell death, primarily due to the formation of hydrogen peroxide. Some evidence suggests that vitamin C generates hydrogen peroxide by interacting with high levels of iron found in cancer cells. Cancer cells often have lower levels of enzymes involved in the detoxification of reactive oxygen species compared to healthy cells, potentially increasing oxidative stress and causing cancer cells to die. It's important to note that successive treatments with high-dose intravenous vitamin C do not increase pro-oxidative markers in healthy people, suggesting that normal cells aren't damaged by the burst of hydrogen peroxide produced by the intravenous vitamin C. Vitamin C also interferes with the replication of viral particles. One in vitro study showed that intravenous vitamin C infected cells that were incubated for 4 days with 150 micrograms per milliliter of vitamin C decreased parameters of virus production such as reverse transcriptase activity by 99% and P24 antigen levels by 13% compared to untreated cells.
Wow.
Another in vitro study showed a dose-dependent effect of vitamin C to kill influenza viruses with a vitamin C concentration of 2.5 millimoles per liter, eliminating 90% of the virus present, and a concentration of 20 millimoles per liter, fully blocking viral replication. A really interesting study using a vitamin C deficient mouse model demonstrated that these mice had increased pro-inflammatory cytokines and a 10 to 15-fold increase in viral titers in their lungs during viral infection compared to normal mice.
Hope you've heard that.
1 week after the mice developed a viral infection, all of the vitamin C deficient mice had died, but all of the normal mice survived.
So